My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. We have a good one for you guys today. I am sitting here with Dr. Sarah Baker. I'm interested to talk to her for a number of reasons. She's worked with a number of species that have always interested me. But, you know, in herpetology, people often kind of get like focused or pigeonholed into certain areas, whether that be like taxonomically, they only work on salamanders, or geographically, or topic-based. And the little I know about Sarah, she seems like a pretty good example of somebody who's made a pretty big jump, in this case, among taxonomic groups, where she had a real focus on snakes. And then as she moved on in her career, transitioned to other groups of reptiles. And so we're going to talk about all the work she's done and talk a little bit about that transition and how it came to be. So welcome to the podcast, Sarah. Good to have you here today.
Thank you so much. Happy to be here. Thanks for inviting me.
Yeah. So I like to begin by learning about my guest. And I like to start at the beginning. And a lot of people in the world think it's very strange what we do and what we're interested in. And but for us, it's kind of normal. So when did that kind of strangeness begin for you? When did you start getting into reptiles? Is this something, from when you were a child, or something hit you later in life? How'd that come about?
Yeah, absolutely. I think I was born with the strangeness. So my parents are not in science. They are people that are kind of on that side that think this is very weird what I do. And we're a little surprised that this was even a job that I could have, right? So since I was a kid, I've been running around, no shoes on, grabbing whatever critters I could find in my yard and building them, keeping them for a couple days, right? Building them little habitats, letting them go later, terrorizing my mom, bringing them into the house to show her what I caught that day, all of that fun stuff. So my parents are not surprised now that I chose this as a career, because since I was a kid running around outside, this is what I've been interested in.
Yeah. You're probably like me then, like at family gatherings, you're with extended family, and it's like, you're like a topic of conversation. Like, it's not, you probably have regular conversations, but there's always a time where it's like, there's a real focused conversation where an uncle or somebody is like, wants to just talk about snakes or whatever it might be, turtles or frogs. So,
oh yeah, absolutely.
So where did you grow up?
I was born in Fargo, North Dakota.
Wow,
so
cold.
Practically Canada, right? Way up there. There wasn't a whole lot to chase around up there. It was mostly garter snakes and toads that I was catching as a kid, which is probably good. There's nothing venomous that could actually hurt me since I didn't really know what I was doing, when I'm 5 years old running around barefoot. But yeah, so I grew up in the Midwest in North Dakota, moved to South Dakota for a little while. And then I always thought I would be a veterinarian, right? I had this interest in animals. I think a lot of people that grow up interested in animals, that's kind of the track that they think they're going to follow. And until I got to I went to North Dakota State for my undergrad degree. Until I got about halfway through that, I still thought I was going to go to veterinary school and be a veterinarian. I've been interested in disease and wildlife health and all of that for a long time. And I'm sure we'll talk about it later. My PhD took a little bit of a turn in that direction anyway. But I once I figured out that I didn't have to be a veterinarian to go to work with animals, right? I was like, research is actually what I'd like to do. So I kind of transitioned about sophomore, junior year of undergrad to the grad school track instead of the vet school track.
Yeah, that's an interesting, from my experience, you hit it, the nail exactly on the head. Like, people like us who have an interest in animals and wildlife and general, like society has kind of taught us, like just as we're brought up, like, oh, one of the major ways that you interact with that is to become a veterinarian. And there's actually a number of guests who have that same story, where when they're coming up, they knew they wanted to work with animals. They loved snakes or whatever it might be, but they were going to become a vet so they could be around the animals. So that's interesting. So, given again, you're kind of like this black sheep of the family, how, when, I'm assuming the vet track kind of was like, yeah, they understood that. Oh yeah, you could be a veterinarian. That makes sense, blah, blah, blah. But how did it go come like freshman, sophomore year when you started that conversation, say, with your immediate family, like, hey, I think I'm going to transition. Was that were they very supportive? Was it, were they confused? How did that go down for you?
Always very supportive. I've had great support, especially from my dad, with my whole career and everything. But he always had said, because I was good at science, right? So it was always, you should go to medical school kind of thing. Mm-hmm. Like, it'd be great to have a doctor in the family. So he accepted the veterinary track as, that's close enough to med school. And then I said, actually, I'm going to go to grad school. I think I want to do research, and I think I want to do research on snakes. And he was a little confused, not unsupportive, but definitely confused. Like, that's a job? I'm like, yeah, it's absolutely a job. He's like, well, what are you going to do after grad school? I'm like, well, there's a bunch of things. I could teach. I could, work for some nonprofits. There's jobs. Like, it'll be okay. And he's like, you'll make enough money doing that? I say, yes, Dad, I think I'll be okay. I'm still going to be a doctor. It's just a PhD instead of an MD or a DPM.
Yeah, I was going to say he has a doctor in the family.
You got your doctor in the family. Yeah.
It's just a snake doctor. Right. Yes. Anyways, okay, well, that's good. I'm glad you had some support as you moved on through that. So I think you said North Dakota State. Is that correct? And
that's correct.
Is that in Fargo? I'm not sure where that is.
It is. Yep.
Oh, okay. Yeah. Fargo. I've been through I've been through South Dakota. I've never been to North Dakota. Yeah, it's cold. What do you have, like 2 weeks a year that the reptiles are out? I'm joking, obviously.
A little longer than that, but yeah, no, it's cold up there. Multiple times it snowed in May during finals week, as I'm walking to take my last exams or snowflakes coming down. Every winter we get a couple weeks blast where it's minus 40 outside. And yeah, it's certainly a narrower window, but out west is beautiful. Shortgrass prairie, prairie rattlesnakes all over the place, bull snakes. It's a gorgeous place.
Yeah. If I remember my geography well, again, there's only 2 states in the U.S. that I have not been to, like, feet on the ground, not airports. One's North Dakota, the other's Michigan. So I'm trying to think of my geography. Is Fargo's like kind of east, maybe east central North Dakota? Is that right?
It's way on the east side, almost on the border with Minnesota.
Oh, okay, great. So are you in the west side? Is obviously shortgrass prairie, like you're talking about. Yep. But is eastern North Dakota, is that tallgrass prairie? Like, I'm assuming heavily agriculture at this point in time, but.
At this point, it's pretty much all agriculture, yeah. But historically, it would have been a longer grass prairie than they had out west. Yeah, the west is more rolling hills, kind of Badlands type areas with the short grass. And it's all, the western half of the state is mostly ranches, eastern half of the state, row crop agriculture.
Ah, okay. Interesting. So you make this decision. You've got some good support at home, and you're going to move on, kind of more on this research track. You're interested in snakes. And so how did you take it from there? How did you find graduate program? How did you end up going to graduate school? Where did you end up going? All of that.
So I took the herpetology class that North Dakota State offered, and the TAs for that class were graduate students that were doing their master's degree there. And one of them hired me as his field tech on his prairie rattlesnake project. So shout out Matt Smith, if you're listening. You kind of changed my life and gave me all the tips for how to get into grad school and my first rattlesnake job. So working with him, I was like, well, how do I want to do this? Like, how do I go about this? And he's like, well, honestly, start contacting people you think you'd like to work with. If you know it's rattlesnakes, just start emailing people whose papers you've read that you enjoy and ask them if they are looking for new grad students. So that's how I found my program. I just blanket sent out a whole bunch of emails to a bunch of different professors asking if they had any availability in their labs for another master's student. And I landed at the University of Illinois Natural History Survey with Dr. Chris Phillips. He had a project going with Eastern Massasaugas there. And I said, I've read a few papers that have come out of your lab. This all looks great. I'm looking for a grad program. And he said, yep, come on out.
That's great. That's a similar, I've told the story before on the podcast, but that's kind of a similar thing. You know, my master's was more, I stayed at the same university I did my undergrad, and I kind of rolled into an existing project. But for my PhD, I did like a very sophisticated, I don't know if sophisticated is the right word, but it's probably giving myself too much credit. But I had a process, and that process was, like created a database. I had my, with you it was rattlesnakes, and I had the things I knew I wanted to work on, and I ended up kind of creating a database, went through all these labs all across the country, had a list of people, and reached out to a lot of people. And I think using a strategy like that can be really important, especially today. I mean, you're younger than me, but I think it's just getting harder and harder to get into graduate programs. There's more competition, fewer jobs, all of those types of things. So going about it in a very dedicated way, like making work of it. So anyways, I had a similar process. But so you end up going to Illinois, and you're going to work on one of the coolest little rattlesnakes in existence. And
I think so, yeah.
And so this is for your master's to start. And so first of all, like thinking about like the project design, I just talked about for my master's, I kind of rolled into a project that was just starting up, but there was big grant funding for it. The proposals were already written. There were kind of, I had a little bit of leeway within that, but there were like very clear goals and objectives relative to the funding agency. So what I'm getting at is for your master's, was it, did you come in? I know this is long-term project that you're coming in on. Did you come in and it was very kind of laid out in front of you, what you needed to do for your master's, or how much leeway did you have in kind of deciding the direction to take your thesis work?
So yeah, I rolled into a project that had already been started in 1999. I think you've had Mike Dreslick on the podcast, and he was the one, him and Chris Phillips started the long-term monitoring of the Eastern Massasauga in 99. So he had just finished up his PhD. I started as a master's student then, and we kind of sat down and had a conversation about what are you interested in? Here's some potential options. And I love to tell, now that I'm a professor, I love to tell students this story. Like what I thought I was going to do for my master's degree, and actually also my PhD, did not turn out to be anywhere close to what I actually ended up doing. So things changed. I was supposed to do a kind of comparison. The state park that we were working in was doing a whole bunch of habitat management. So it was supposed to be wet prairie down there. They had a lot of woody vegetation encroachment from kind of not managing it as prairie for a number of years. So the plan was they had these huge areas of habitat that the snakes did not use because it was too close canopy. They were going to clear cut all of that, and I was going to monitor how long it took to start finding snakes using the restored habitat. Well, if you've ever worked with state government, things get delayed. They don't happen exactly when they're supposed to, through the fault of not necessarily the staff, but just things get delayed. So that didn't work for a master's timeline. They ended up doing the restoration work, but it was not going to be in a timeline where I could do a master's project on that. So I kind of switched angles completely, ended up doing a physiology project and measured metabolic rates working with Steve Beaupre at the University of Arkansas.
Who's also been on the podcast.
Who's also been on the podcast, yeah. So Matt Smith, the grad student at NDSU, was a PhD student with Steve Beaupre when I went down there to make some measurements on the Massasaugas for my master's work. So rattlesnakes is a small world, we all know each other. So everything kind of came full circle there. But so, yeah, I switched completely and drove a bunch of snakes from Illinois to Arkansas to make some metabolic measurements in Steve's lab.
You didn't switch labs up, like you didn't change universities and then go to Arkansas.
I switched projects. No, yeah. I was still a University of Illinois student, but Steve allowed me to come down and use all his lab equipment for those measurements because we didn't have that equipment in Illinois.
So was this new kind of metabolic focus for the masters? Was this also was it within the framework of this restoration project, meaning like maybe like modeling potential metabolic rates, whatever, thermal ecology in, say, existing good habitat versus existing poor habitat, predicting what was kind of the framework, or was it just purely to understand the physiology of the species?
We had kind of tossed around a bunch of different places that we could potentially expand the initial work, but for just the confines of my degree, no one had done the baseline metabolic measurements on these guys before. So I brought everything from very small juveniles to very large adults to measure and to just get the baseline resting metabolic rate for that project.
So, so our audience is very diverse. There's some scientists and, physiologists and taxonomists, and there's, carpenters and, just a wide variety of people. So, first of all, just give you're talking to your grandmother about what is metabolic rate, and then maybe kind of talk to us about how you measure that in a snake, or how you specifically got to that resting metabolic rate with Massasaugas, almost like the techniques there.
Sure. So metabolic rate is essentially how much energy are you using to stay alive. There's a bunch of different metabolic rates you can measure, kind of how many calories you need. So we, as mammals, have very high metabolic rates because we are using a lot of energy to regulate our body temperature. Reptiles are extremely interesting, and venomous snakes in particular, because they have extraordinarily low metabolic rates. They don't move very much. They're not wasting a bunch of calories. Keeping their body temperature regulated. They do all that behaviorally with moving in and out of the sun. So we were kind of coming at this from an angle of how many calories do Massasaugas need just to stay alive? And we can kind of equate that back to rodent populations at our study site. So that was kind of the idea that we played around with. So to do that, I packed up all these snakes, drove them to Steve O'Pre's lab. We put them, what we want to get is just the minimum amount of energy for them to stay alive. So we don't want them to be able to move. We don't want them to be active. So you put them in teeny tiny little plastic containers and stick them in an environmental chamber that you can set the temperature and you can set the lights so that they'll have a normal day-night cycle while they're in there, and hook up a flow of oxygen. So what we're essentially measuring is how much oxygen they are consuming while they're sitting in that box. And that will give us an idea of how many calories they're expending with some models and some equations that you do on the back end there.
Gotcha. And do you see, just so those base rates, do you see, how does the variation break down? How does it, do you see differences among sex or size or, condition of the animal? Like, did it, I don't know, eat, not eat? What are the factors that influence that? Or is that kind of base metabolic rate kind of the same across all of these potential factors?
So what we see is that metabolic rate will vary in a predictable way by body size. So very small individuals are going to need less energy than very large individuals. And we can plot that. And that curve is very similar for a lot of snake species. And then if you have a gravid or pregnant female, she's going to be using more energy, dealing with all of the caveats of that. The very interesting thing that I found when I measured the Massasaugas is we did have a significant difference in resting metabolic rate between males and females, which
independent of like whether she's gravid or not. So a non-gravid female.
Of males and non-gravid females. Yeah. Yep, independent of that. We made sure all of our females were not gravid. So Steve had never seen that before in any of the species that he'd measured. So
So males were
he was
higher.
surprised. Males were higher, yes. Yep. So that was a surprise to us. We were like, oh, that's interesting. Steve thought for a minute that we might have made a mistake somewhere when we were collecting the data, but we went back, we reanalyzed it a few times, and it's a real difference. So when we later on, we kind of went back to some of Mike Dreslick's data that he had collected and analyzed growth rates, and males have an extraordinarily faster growth rate. They reach sexual maturity a lot faster than females. So we kind of think we figured out we can attribute it to the very fast growth rate that we see in males compared to females.
Yeah, so I'm sure you've speculated in your mind or maybe in the discussion section of a paper that I probably haven't read, but like, why would that be? Do you have any like speculation on like why would it be advantageous for males to have this higher metabolic rate, get larger faster? What are your thoughts there?
Hey everybody, I just wanted to take a quick break and tell you that the Orianne Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals, but these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.orianne.org to learn more about our work and how you can get involved.
This is purely speculation because we never could figure out a way to do follow-up studies to prove whether we were right or not on that. But the Massasaugas in the wild just don't have that long of a lifespan. In captivity, they can live a very long time, but it's pretty rough out there. And a 6-year-old snake is pretty old for the population that we were working in. So evolutionarily, if you're a male and you can put a bunch of energy into growing really fast, and that gives you more seasons to be reproductively active to increase your fitness and the number of babies that you can make, right? Yeah. So we think that's why, and it's just not as energetically costly for males to mate and reproduce as it is for females. It's a lot less on them, so they can mate every year. If they get big fast, they can get a few active seasons in there, reproduce a number of years in a row before maybe they don't make it to that 7th or 8th year.
Yeah. Huh, that's interesting. I forget, with females, that's a short life for a rattlesnake, looking among species. So like for females, when do they, I should know this, and I'm sure I've had it on the podcast because I've had multiple Massasauga episodes. But how old are females when they become sexually mature typically?
In our population, it was usually around 3 years old.
Wow. So she becomes sexually mature. And did they breed every year, potentially, the females?
Every other year.
Yeah. So a female is giving birth once, maybe twice, if she's lucky in her lifetime.
Exactly.
Yeah. Very interesting. How about more speculation here, which I know scientists don't like to do, but so bringing it back to kind of the applied side of things. So you have this metabolic rate. You have this really interesting result, between the sexes. You've kind of speculated as to why that might be relative to their life history. And then, but you have this kind of applied question that was originally going to be, part of your thesis, the focus of your thesis, this idea of, prairies, native prairie is one of the most endangered ecosystems on the planet, in particular the tallgrass prairie. And so, any speculation about metabolic rate relative to these different kind of habitat types? And what I'm getting at is this non-prairie habitat type, this forest that comes back if you don't manage prairies like they would have been prehistorically and historically, do you think metabolic rate is one of the keys behind that relative to these habitat changes as to why they decline, don't live there, or do you think it's other things?
I think they don't live in the forested areas because they don't like canopy cover. We had done a lot of telemetry at our site, and we just never found them in the areas with canopy cover. Also, the structure of those non-restored areas was basically like invasive autumn olive trees and bare ground underneath it. So there's nowhere for them to hide. They don't like to be exposed. So I think that's why they were not in those areas. But one of the things we did with my metabolic rate calculations is kind of get a number. How many mice does a snake need to eat in a year just to meet baseline requirements? And we were using that to try and figure out, we were going to do some small mammal trapping, which again is one of those side projects that you never quite get around to when you're a grad student, right? We were going to do some mammal trapping to get estimates on the different mammal species that were out there to see if one of the reasons the population wasn't increasing was because there just simply wasn't a high enough prey base. So we were kind of looking at this idea of carrying capacity. Like, is this particular site supporting as many snakes as it can based on the size of the rodent population? Because especially the smaller Massasaugas are really, the juveniles in particular, are really only eating southern short-tailed shrews at that particular site. That's the only mammal that's really small enough for them to swallow at that juvenile stage. And we did some feeding trials. They were not interested in eating lizards or frogs or anything else. They really are mammal specialists. So that was kind of the idea there, and the angle was, can we figure out if we're at carrying capacity and all of that based on mammal populations?
Very interesting. Okay. So you do kind of this somewhat novel research for your master's on metabolic rate, and find some things that are kind of new to science. And then, it comes time, your master's is kind of wrapping up, and you've got to make some decisions in your career. You've already mentioned that you were there for your PhD as well. Was it just kind of an automatic thing? It was like, oh, I'm staying here and going to continue working in the system, or, did you, really think about it and just, am I going to go get a job? Am I going to go work in a different system? How the transition to master's to PhD, how did that look for you?
Yeah, for me, it was automatic. I had no interest in leaving. Fell in love with the little Massasaugas. They're great. That study was just amazing. The amount of long-term data that we had that we could pull from. So I basically finished up, before I finished even and defended my master's, I basically said, I would like to do a PhD here if you guys will let me stick around. And Chris Phillips and Mike Dreslick were happy to have me around. Weren't sick of me yet. So I stuck around and did a PhD on that same population as well.
And did your PhD work kind of build off of your master's work, or did you kind of go in a new direction? Basically, kind of how'd you design what you're going to do for your dissertation, and what was the focus there?
So I was still very interested in conservation, and what I had wanted to do in the first plan, I wanted to focus on what was going on spatially with the juveniles. So we had done a lot of telemetry, but always on adult animals. And no one really at the time when I was reading the literature, not a lot of people had spent much time doing any kind of spatial ecology on neonates, right? So right after they're born, what's their overwinter survival rate? How do they decide where to hibernate? Do they hibernate with mom? Do they hibernate with siblings? So that was kind of what the focus was going to be. I bought the absolute smallest radio transmitters I could afford at the time, which weighed 1 gram. And they were only going to last. I slowed the pulse rate on those transmitters. Anybody that's done telemetry, the more beeps that you have per minute, the easier it is to find your animals. I slowed mine down to make the batteries last as long as I could, all the way as low as 7 beeps per minute, which is very slow. And and really had an interesting time following all these little snakes around.
I was going to say, you must have done circles in these wetlands, just been like, where am I? Where's this snake?
Luckily, they don't move very much, right?
Yep.
So they're not making these huge long distance movements like a very large snake might. So when I got to their last known location and I was tracking them every day, they usually hadn't moved very far. So that was what saved me there. But yeah, it was a challenging telemetry project, to say the least.
And did you,
but because they were so small,
well, I was just going to, I'm sorry,
go
maybe that's where you're going. I was going to ask about where you attached or implanted the transmitters.
Yep, that's where I was going. So they're so small. The snakes are so small, they're endangered. This is the last population that was in existence in Illinois. And they would not let me do surgical implantations due to some of the risks to the snake themselves. So I attached them externally with glue, which was also very challenging to keep them on for any long period of time, moving through tall grass prairie habitats with a transmitter glued to the outside of the snake. I lost the transmitters came off. We'll just
so you glued them
so.
to the body, or Massasaugas, if people don't know, they have really tiny rattles, but a number of people, and we've done some of this, attaching transmitters to rattles now, but I would think in a Massasauga that'd be almost impossible.
Well, in an adult you could do it, but in the neonates with just the little tiny button that I was trying to track, it was too big to glue to the rattle. Yep. So I kind of glued them to the side of the body, which was the best place that we all sat down and tried to figure out how we were going to do this. And that seemed like the best option. But
and what happened when they shed their skin? Did it just come off, or did it kind of like...
Yep. Okay. It did. So the plan was to watch them, get visuals, and when they started to look like they were going to shed, the eyes, for anyone that doesn't know, the eyes of a snake get cloudy and look a bluish color when they're about to shed. So I would pull them when they were about to shed, let them shed in captivity, stick the transmitter back on the fresh skin, and put them back out. Spoiler alert, this did not work very well. So if anybody listening is wondering why they never read this paper, it's because I didn't have a high enough sample size at the end of the year to even be able to publish any of this, because transmitters fell off. And turns out the snake's skin got very irritated by the glue and they would just shed constantly. So I like to tell my current students this story too. Not all research projects work out. Like sometimes you put a lot of time and effort into something and it doesn't work.
Yeah, I mean, that's what I was wondering, like when they get this thing glued to their side, if then that kind of makes them want to shed their skin and they go into a different cycle as well. But I mean, you're obviously up for a challenge, because the ecology of like neonate or juvenile snakes really across the board is one of the big holes in our knowledge in snake ecology, and you went and picked one of the smaller rattlesnakes to try to do it. I tried, way back when I did my dissertation on Great Basin rattlesnakes, I tried to do the same thing with neonates and, was equally unsuccessful. So anyway, so you have this great idea. Technology is not there yet to answer it. So sounds like similar to your master's, you needed to pivot. So how'd you pivot?
I did. So at that point, I kind of tried this for a couple of years. We realized, we're just not going to have enough. I mean, I got some interesting data, but the sample size was just not there for me to be able to kind of write this up as a dissertation and get publications out of it. So we kind of shifted away from the focus being on that. I was always interested in conservation of these things anyway. So dissertation turned into a chapter on sources of mortality, a chapter on population genetics, and a chapter on some updating some reproductive ecology with litter size, because to get the neonates, I was catching gravid females and letting them give birth in captivity anyway. So I had all this data on litter size and all of that. And a previous student of Chris Phillips, Ben Jellin, had done some reproductive litter size work before. So I kind of added some data to what he had done. And then a population viability analysis was my final chapter, to see, what are the chances that these things will persist for the next 50 years.
So let's talk about a couple of those. We've talked about the genetics there in the past. So first, what was the first one? To me, it was one of the most interesting.
Sources of mortality.
Yeah,
so we kind of...
Tell me about that. What's killing these Massasaugas? Or how are they dying?
So,
would be a better way to put it.
The most interesting part of that analysis turned out to be, depending on what method you use to collect the data, your results on what the biggest threat will be different. So what I mean by that, we had a ton of telemetry data. Mike Dreslick did a lot of telemetry for his PhD. We had a ton of data there. And then we also had kind of a separate data set, if you will, of snakes that we encountered that were dead, doing visual encounter surveys, because we were out there every day during the spring emergence period looking for these things, driving on all the roads around the study site. So if you use the telemetry data only to decide what is the biggest threat, predation is the number one source of mortality there. We have prairie king snakes at the site, which are known rattlesnake eaters. We have racers at the site, which I tracked when I was doing the telemetry on the neonates, I tracked 2 of my neonates to the same blue racer that had eaten both of them. I tracked a transmitter and found it in a screech owl pellet. So there's predation. Lots of things eat these poor little rattlesnakes. So telemetry-based studies will find predation is the biggest source of mortality, and visual encounter-based studies will say that roads are the biggest source of mortality.
Interesting. Okay, well, how about some of these updated reproductive metrics? What did you guys learn there? It sounds like litter size was a focus. So maybe talk about what you learned, what you added to our knowledge of reproduction, and then you can transition into what that meant for your viability component.
So kind of what we tried to do with that, since we had all of this data on, when the females give birth in captivity, you get the live young, you get the sizes, you get the stillbirths, you get the ones that they pass that are undeveloped eggs still. But all of that kind of is a calorie investment, right? Females spend a ton of calories investing in eggs. They lose 50% of their body weight after they give birth. So we actually did Mike Dreslick helped me put together this kind of survival model, the same way that you would model survival of living individuals. We modeled sort of survival from the egg to birth in that way, and to look at sort of wasted caloric effort, if you will, on how much of the energy that they put into developing these eggs actually never produces live young. So that was an interesting part of that we looked at. They don't waste much. They have a pretty efficient reproductive system. And then we also kind of looked at, the thought just left my head what I was going to say.
Well, tell me about viability. Are these, is this one remaining population going to be there in 50 years, 100 years? What are we looking at?
So if nothing changes, it looks like the population should be able to hang on in small numbers for the foreseeable future. It wasn't, I kind of expected more of a crash as far as the viability goes, because it is a very small population and has been for quite some time. But as the population genetics that I did, and that I'm sure some of your other guests have talked about with this species, shows, they actually do pretty well in small populations, probably because they utilize multiple paternity. So a female can have her litter fertilized by multiple different males. They'll mate with multiple males in one season. That keeps the level of genetics pretty high, keeps them from getting inbred. And they're doing okay, but whenever you have small populations, any one catastrophic event could take out a number of reproductive age females, and that's when you start to see these crashes.
I know that snake fungal disease has been pretty prevalent in Massasaugas, and I think it's one of you can correct me if I'm wrong, but I think it's one of the species where actually we've kind of seen an impact of the fungal disease, so going beyond prevalence. Do you have it there? Is it an issue? Could that affect this viability prediction?
Yeah, so that turned into a little bit of a side quest while I was doing my PhD, because I was out looking for gravid females one summer and found 3 snakes that had swollen, absolutely messed up faces. And being as though I had a previous interest in wildlife health anyway, since I thought I was going to be a veterinarian for a long time, called up a wildlife veterinarian that had been done some undergrad research with Chris Phillips, named Matt Allender. He's a good friend now. We started working on this. I said, I've got these snakes. They don't look right. Do you want to look into this with me? And it turned out those were the first cases of snake fungal disease that we saw at the site. And so then we started annual sampling, looking for it, prevalence rates, all of that fun stuff. Found a lot of snakes that looked really messed up. A lot of snakes that tested positive one year and we never found them again. So did they die? Did they just move somewhere? With some of those visual encounter surveys, it's always hard to tell. But I don't think it doesn't have an impact. It doesn't seem like at first it was kind of major panic, right? Like, oh my God, this is the nail in the coffin. All of these snakes have these absolutely horrible fungal ulcers. Like this is going to be what kind of kicks the population down the extinction vortex, so to speak. I don't think it is going to be that, but if a female misses a reproductive season because she's covered in fungus and doesn't mate or whatever, that's a big deal for these small populations. So I can't imagine it doesn't have an effect, but I think our initial panic at the site of this is going to be what does them in is not going to be what happens.
Yeah, another thing just hit me thinking about like insidious factors, things that just kind of spread and you can't control in a conventional, deal with it in this place in this way. Down here, as you may be aware, we're dealing with this parasite that was introduced from, came with the pythons, we believe, and is moving up Florida, is, probably moving into Georgia as we speak, and it's impacting snakes. And one of it's probably impacting multiple species, but the species that it's been studied the most is pygmy rattlesnakes, which is obviously closely related to Massasaugas. And they're seeing real impacts of this parasite. And it just hit me while you were talking about the fungal disease. I wonder, first of all, this parasite is moving very quickly across the landscape. Like, I don't know if it could survive up there. Somebody probably does. I'm probably speaking from ignorance here. But yeah, I wonder if that's another insidious type of threat that in the future need to worry about.
Hey everybody, I just wanted to take a break and tell you guys that snakes are one of the most persecuted groups of animals in the world, but we are dedicated to changing that. The Orianne Society works every day to ensure there's a future for these amazing animals. If you care about snakes, wildlife, or wild places, and you like what you've been hearing on the Snake Talk podcast, support us today by donating on our website at www.orianne.org.
Yeah, if that, the pentastome worms is what you're talking about, right? If that
Correct.
Gets up there, I can certainly see how that could be a problem as well.
Yeah. Anyways. Okay, so you are living your dream. You're not a medical doctor. You're not a veterinarian. You're in the field. You're working with these snakes, these animals that you always wanted to work with. And then you went and made this strange decision that you're going to go work with turtles. But what I'm getting at is you finish your PhD. And you move on to your career. Kind of what were the steps that got you to where you are today, and where are you today? And then, if you can talk a little bit about that transition too, because it sounds like it's not that you went there and there was a snake project, so you did it, but you love all wildlife, and you could have been working on flamingos and you'd have been happy. You were working on snakes, you were all in. And now your research programs are in the reptile world are more focused on turtles. So talk about how you got to where you are today from your PhD, and then let's talk about that taxonomic shift.
Yeah, so after I finished my PhD, I stuck around for another couple years at the Natural History Survey as a postdoc, and saw a job posted to go work for the Arizona Game and Fish Department. And as a rattlesnake person, I thought, let's move to Arizona. There's more rattlesnakes there than just about anywhere else. Let's go do that. I moved to Arizona for 2 years. Working in state government was not all I thought it might be cracked up to be. That job didn't work out for various reasons. And I also missed academics. I missed the academic research environment. It's working in state government is a lot different than that. So started looking for faculty positions. I thought, I miss teaching. I liked teaching more than I thought I would when I was a grad student. I thought, I'm going to hate being a TA. I don't want to do this, but I actually really enjoyed it. And I missed that. So I started looking for academic positions in areas where there would be a high diversity of reptiles. And I knew I wanted to work at a smaller school. I absolutely loved my graduate experience at the University of Illinois, but that is a big school. And for being a faculty member, I was more interested in smaller schools where you kind of smaller class sizes, more chance to interact and actually get to know your students than some of those bigger universities, the R1 universities. So I was looking at small schools and areas where there'd be a lot of reptiles and amphibians, and found a position at McNeese State University in Lake Charles, which is where I am now and have been for the last 5.5 years.
A little bit different weather than Fargo.
I was born and just started moving south and never stopped. Yeah.
Great. So you ended up doing a little stint in government, and then got into academia. And I'm assuming McNeese, you have a pretty heavy, like, teaching's a big component of what you do, but it sounds like
it is. These smaller schools, all of them are like, it's a very much teaching focused, which I do enjoy, but so still doing research, but it is a teaching focused position.
So you're a snake person, and you probably realize this, but you're in Louisiana. There's lots of snakes there. So tell me, how did you get to turtles? It doesn't seem like you were into snakes and you're still doing, it was like snakes, now turtles. So tell us that.
Well, I haven't completely abandoned the idea of snakes. I was really interested in trying to do some pygmy rattlesnake stuff here in Louisiana, but it's a little bit different of a habitat. So there's so much area and they're so spread out, and it's very marshy down here. I'm in the southwest corner of Louisiana, very close to the coast, and I don't have a boat, right? And I don't know how to drive a boat. So getting around some of these places, there's snakes everywhere, you're not wrong, but getting around some of these places is challenging. And there's an overwhelming number of water snakes just everywhere. Yeah. So I did start a bit of a project working with another professor down here who's actually in the chemistry department, but is interested in reptiles, Mark Merchant, who has done a lot on alligators. When I got here and we started talking, he's like, well, let's do a snake project. And I said, great, that's obviously. So we started this project going out. I was really interested in cottonmouths, honestly. And we just caught 1,000 water snakes and 10 cottonmouths at this site that we were working at. And I don't have anything against water snakes, but they're not my favorites. They're very bitey. They don't smell great, right? Yeah. So I kind of was like, well, this is proving to be a little bit challenging, but I know I can set traps for turtles, and there's turtles everywhere. So we have a bayou that runs right through campus. I have undergrad research students that have gone out, thrown turtle traps in the bayou. We catch 1,000 red-eared sliders and occasionally a snapping turtle or a softshell. And then Mark and I were talking about, well, what other turtles could we kind of do some research on down here? And since we're on the coast, Diamondback terrapins were kind of a natural thing to flow into. They're very cute, fun little turtles. Also potentially some population level threats going on there. So we started a mark recapture project on the terrapins, and I've kind of been a little bit more focused on that.
Talk about, so what is, for our audience, what is a Diamondback terrapin?
So terrapins are interesting. They're the only species of turtle that prefers brackish water habitat. So as opposed to the sliders and the soft shells that I catch on campus, those are freshwater turtles. They don't like it when it gets too salty. Sea turtles are completely saltwater species. They don't spend a lot of time in fresh or brackish water, but the terrapins are found pretty much just in that little narrow band of brackish water marshes that we see on the Gulf Coast, and then going up north on the Atlantic coast up to about Massachusetts.
Okay. And how are the populations? I mean, obviously they're kind of a conservation concern throughout their range. I say obviously, but they are. And
they are.
And you're kind of over on one end of the distribution. What's kind of their status in, the marshes of Louisiana?
It seems to be, the population seems to be pretty large. We haven't run all the population size models on that data. That's kind of an ongoing project that we've got, but we've captured quite a few individuals. I think it's up around 500 individual turtles at this point that we've marked from this one population that we're working at. So it seems like they're doing okay right there. There are some pretty substantial threats. Crab traps is one of them. When you fish for crabs, oftentimes you will bait traps, sink them to the bottom, and the crabs will go in there, and you'll go in and collect your crabs, the next day or something. But the turtles will swim in there to eat the crabs or the bait as well, and can't breathe, obviously, and end up drowning in crab traps.
Don't they have that's a crab trap, or turtle excluder devices essentially for crab traps, or is that not the case?
Commercially, yes, but private individuals are not required to use the turtle excluders.
Ah, interesting. Can they eat the turtles if they catch them and they die?
No, they are protected.
Okay.
So you are not supposed to harvest them.
Yeah, because obviously, Diamondback, I say obviously, I keep saying obviously and then saying something. It's not obvious. But they used to be heavily, hunted or trapped for their meat. People think of turtle soup. Like historically, that was one of the major turtles that you'd find in turtle soup. So, and in it, I was just curious because then it would give them an incentive to catch more of them if they could keep them and eat them. Okay. Well, what does the future hold with that project? It sounds like part of it's it's educational as well. It's experiential opportunities for your students. So the continuation of that, do you have any kind of directions you want to take that in the future besides just kind of getting the mark recapture data and ultimately being able to estimate population size and survival and whatever else you might do with that?
We do. We want to go kind of in a spatial ecology direction and see if we can figure out where they're nesting. The nesting areas at the particular site that we're working on are not obvious of where I would think turtles might go and lay eggs. So I'm very curious if we can figure that out. We're working on a diet study. So female Diamondback Terrapins have very large heads. And when you compare the size of the head of a female terrapin to a male terrapin, there's an obvious difference there. You can pretty much sex them just by looking at the size of the head. So what implications does that have on what they're eating? So we're kind of looking into some diet related issues there. And are the females consuming more crustaceans? There's a lot of available food. There's fish down there. There's crabs. There's snails. And is that head size difference? Females have more muscles, more jaw muscles. We are going to measure bite force, see what the force that they can produce with those large jaw muscles, see what implications that might have for different things females can eat versus males, and kind of take it in that direction.
So I'm guessing you have a boat and know how to drive it now if you're working on terrapins.
Well, my collaborator has a boat now.
Okay.
Also, we can access these sites with kayaks, so
okay.
I don't have to drive the boat.
Got you.
I still can't drive a boat. My own detriment, but
you live in southern Louisiana and can't drive a boat. That's crazy.
No.
Anyways, okay. Well, so I also mentioned earlier in the episode that you're doing some box turtle work. I'm assuming it's kind of similar in that, it's kind of experiential opportunities for your students. But talk a little bit about that before we wrap up. What are box turtles like in Louisiana? I just picture the whole state's just a big swamp. Like, where do the box turtles live? And
yeah, so we do have some upland areas.
Okay.
Box turtles, eastern box turtles, are kind of declining through a large part of their range for various reasons. I don't think all of them have quite been picked out yet, but we have box turtles here. They're very charismatic. From a biodiversity perspective, if you've got a nice healthy habitat, you should have a nice healthy population of box turtles. So I'm going to try to get some students involved and start some spatial ecology, radio telemetry stuff with the box turtles, and just try to look at do mark recapture, population size, where are they going? If we can, this is way down the road because I'm kind of just getting this box turtle stuff off the ground, but if we can figure out some nesting success, because one of the big drivers of turtle declines is nest predation. Turtles go and lay their eggs in a little hole in the ground, cover it up and leave, and then raccoons, possums, skunks, all the little mesocarnivores can sniff those nests out pretty well, and a large number of turtle nests are dug up and all the eggs are eaten within 24 hours of them being laid. So that's an across the board problem for turtles. So we're going to way down the road, see if I can get some students interested in doing some box turtle nesting surveys and see if we can't figure out what the nest success rate is here in our little corner of the world.
Interesting. So do you have any other big taxonomic shifts in your future, or are you pretty set on these turtle projects as long-term and kind of the cornerstone of what you do with your students, or do you have other things on the horizon beyond these turtles?
Those are, kind of what I'm thinking right now. I did just get back from helping a colleague with some bat research in Barbados, so you just never know, right? Just try to stay away from the mammals, though. That should probably be a one and done. Don't want to spend too much time with the endotherms. But.
Cool. Okay, great. Well, I enjoyed our conversation today. As you know, I like to have all of my guests tell me a good snake story. So, Sarah, what's your best snake story?
I'm going to go with a snake story from when I was a kid. Again, so we're going back to Fargo. I'm running around catching stuff all of the time. I've got two younger brothers. Let's see, set the stage. I think about 7 years old in this story. I've got two younger brothers, and the kids that live across the street from us are not particularly nice to one of my brothers, right? So I'm the oldest, being the good big sister that I am. I'm going to teach this kid a lesson that's been bullying my little brother, right? So I happen to know that this kid that lives across the street is not a big fan of snakes. So my brother comes home crying. I go out into my backyard, catch the first biggest garter snake that I can find, march myself across the street, get him out of his house, and start chasing him around the neighborhood with this garter snake, terrorizing him. He never messes with my little brother again.
I love it. You're a snake bully. That's great. That is great. That's a first. We've never had a snake talk story like that. So I love it. Anyways, well, really appreciate you joining us today. I enjoyed hearing your story and all the things that you've worked on and are continuing to work on. So hope you keep doing what you're doing.
Yeah, thank you very much for having me.
Great. And I just wanted to thank the audience and tell everybody to remember, snakes are animals too, and it's a privilege to see one in the wild.